text, and in order, thank goodness, right? Because up until this point, I haven't really been following your textbook in order. But at this point, you'll be able to follow me a little more closely with these. Epithelial, connective, and then we'll look at the three types of muscle tissue and talk about the differences in their anatomy. And then we'll end up talking about nervous tissue. And I'll give you a couple of examples of that. This one we'll spend the least amount of time on. These two will come in much more glorious detail. This presentation is intended to emphasize the majority of our time will be spent on the epithelial and connective tissues. All right, so epithelial first. We say that epithelial tissues are cellular in nature. They have a cellularity, that is, they're composed almost entirely of cells. Now, this is where teachers come in, right? Clarify what you mean by this. Textbook tells you that this is a characteristic of epithelial tissues, their cellularity. But you learned very early in your life that all of the body is made of cells. In fact, we are comprised of about 300 trillion cells. So in what way is epithelial tissue special in terms of its cellularity? And the answer to that question is, we are going to define the tissue by the shapes and sizes of the cells that make it. It is, in fact, as a tissue, cells. Let me just give you an illustration by what I mean by this. It is, as a tissue, cells. So whenever we look at an epithelial tissue, it will be made up of completely the cells for which it is named for. So the cells are flat, or they're cubed, or they're columned. There's one layer or two layers. It is defined by its cells. Now what we'll see as we move into the connective tissues is they are not defined by their cells, at least not structurally. They are defined by their matrix, primarily. So as we get to connective tissues, what we're going to see is there are, in fact, cells there that make the tissue, but they are spread out in the tissue. And we're going to define the tissue with the matrix that's between the cells. All right? The second thing, and I have a little blurb to say something more about this in terms of contacts on the first epithelial slide, the first example. Specialized contacts. They form continuous sheets held together by tight junctions and desmosomes. Now, if you had a good anatomy teacher, you would already know this. You would already know what a tight junction and a desmosome is. In fact, I did that on purpose. Do y'all see the reason now? I taught you what a tight junction was, and I taught you what a desmosome was, the protein nature of those. And I'm going to do it one more time, and I'm going to add junctions within the endothelium of our bodies. Very important to mention, I think, early on. With these specialized contacts, I'm going to point those out to you one more time. All right? Third, very important characteristic. They're avascular but innervated. To say something is vascularized or avascular means it either has blood supply or it does not. So, epithelial tissues are avascular. What that means is, if you look in a microscope at a tissue and you see a blood vessel there, that is not epithelial. Now the question becomes, how do you keep epithelial cells alive? If the tissue does not have blood supply, how do you keep it alive? And so what is important for maintaining epithelial tissues, and we'll see it in all of them, are the basement membranes, the content of the junction membrane between the epithelial lining and the underlying connective tissue associated with it. So we'll learn that all the connective tissues are vascularized. Where does the blood supply, where does the content that is needed by the cell to maintain itself, to stay alive, come from? Normally, it comes from the underlying connective tissue through the basement membranes. It flows out of the vascularization in the underlying connective tissues normally. The second one is the word innervated. To say that something is innervated is to say that it has nerve supply. So to say that some structure is innervated and it has nerve supply would be redundant. When you hear the word innervated in anatomy, you should think nerves. The nervous system. It's innervated. That means the nervous system. Last characteristic here is these tissues are highly regenerative. That is, cells are rapidly replaced whenever they are lost. The easiest illustrations for this are the linings of the insides and outsides, the lining of the insides of the tubes within us and the outside of our skin. So the epidermis of our skin is actually epithelial. And the entire layer of epidermis in most places on our skin, from the basal layer all the way up to the corneum, is replaced every 30 days. So millions and millions of cells being lost over that time period and replaced. So the inside linings are also this way. And one good illustration that helps students remember this, I find, is there's a very interesting transition in the epithelial lining from the esophagus into the stomach. The stomach has mostly a columnar epithelial lining, and in the esophagus we have a stratified squamous lining. And we know that these stratified squamous linings are really highly regenerative. And so people with stomach acid who are constantly moving acid up into the esophageal, through the esophageal sphincter into the esophagus, tend to eat away at that epithelial lining. And the cells respond to that by producing more cells. Now, this is a very common way that esophageal cancers begin, by the overproduction, the regenerative nature of these cells, overproducing them, creating tumors in the esophageal wall. So they're very highly regenerative, lots of replacement. Okay, as I said, we define epithelial tissues based on, on their cells. And this is a cartoon to illustrate that. Not only do we define them based on their cell shapes, which is the next picture, but we base them on the number of cells in the lining. And we have only two varieties of this. We say that an epithelial lining is simple if it has one layer. We say that it is stratified if it has multiple layers. Now there's one more that we'll look at, and we've already seen it. I've showed it to you once already. There's one more way of defining these tissues, and that is there is a very important epithelial tissue that lines our respiratory system, most of it, that looks stratified at first glance. But at high magnification and close inspection, you realize that the cells actually extend, every cell actually extends from what we call the apical surface. So that's an important anatomy term. The apical surface, that is the surface of the lining closest to the lumen. We're talking about the inside. This apical surface is the surface on the outside you can touch if we're talking about the skin. The apical surface is the surface away from the basal surface, sometimes called the basolateral surface or the basal surface. The basolateral or basal surface is the surface on the other side. That is the one where the basement membrane is. It's going to dictate the flow of nutrient into the tissue. So an apical surface and a basal surface in some of the cells that I'll show you for the respiratory system, you will see that it looks stacked, but it's not. In fact, the apical surface of the cell can be traced within the cell all the way to the basal surface. And so it's actually not stratified. And so those tissues are called pseudo or false, pseudo-stratified. So we'll point those out as we get to them. Simple, stratified, and then the pseudo-stratified cells, which we'll illustrate for you. All right. Now we define each of these also with the shape of the cells that make it. So this is what I meant by cellularity. The cells are flat, they are squamous. They are cuboidal, they are cube-shaped. They are columnar, they are column-shaped. And now we can build variations on each of these for epithelial. We're going to have a simple squamous and a stratified squamous, and a simple cuboidal and a stratified cuboidal, and a simple columnar and a stratified columnar, and a pseudo-stratified columnar. So You just put combinations of these words together and you describe your epithelial tissues. Very simple. Okay, now the complexity becomes, the complexity comes in learning where these tissues are and what they look like. So let's use some illustrations in order to do that. This is the first one, simple squamous epithelium, and this is a photomicrograph of part of the lung tissue. So these are alveoli. And as you can see in this image, the alveoli, these little bags where air exchange is going to take place, are lined by these very thin layers of cells that are connected to each other. And those cells are in fact simple squamous epithelial cells. Now, you can see I've made myself some notes over here so that I don't forget to say what I'm supposed to say. With each of these slides, as we look at them, the important part of the slide for us in anatomy is not going to be this picture, this part. The important part is going to be this part. So both in lecture and lab, I'll expect you to be able to describe what the tissue looks like, be able to recognize it, but then be able to talk about where it's located. Because that's really where the business of human anatomy and then ultimately physiology will be, recognizing where these tissues are. Okay, so let's talk about some of these. I've just mentioned the air sacs of the lungs, the alveoli of the lungs. We also know that simple squamous epithelium is in the kidney glomeruli. Now, at least for me, I know Dr. Criswell probably mentioned it briefly to you. It is, if you stay with anatomy and physiology, the kidney glomeruli are going to become very important in your life on more than one occasion. This is the capillary bed. This is the capillary bed in your kidneys, where all of the fluid that is going to be transitioned into urine crosses into the kidney tubes. So it's an extremely important location. And the anatomy of the glomeruli point out first, so I just mentioned this is where the transition occurs. They point out first that these are actually blood vessels. These are capillaries. And so this gives me an opportunity to tell you that capillaries are in fact simple squamous epithelium. Now you can see that's coming more here, right? I'm going to talk about these vessels more here in just a moment. So as we learn simple squamous epithelium, we always want to think about the cardiovascular system. As it turns out, blood vessels, whether it's arteries or veins, have 3 layers. So they have 3 layers. So I'm going to, this is important to us. We don't want to get away from the first one without learning this anatomy. Blood vessels have 3 layers. The inside lining of blood vessels, whether it's an artery or vein, is simple squamous epithelium. Now, as we proceed through this presentation, I'm going to bring that up again, because the second layer of blood vessels is smooth muscle. So if you're going to control blood vessels, the sizes of their lumens, you do it with the middle layer, the smooth muscle. And the outside layer is connective tissue, mostly loose areolar, and so that's going to come up again as well. All right, so this gives me an opportunity to introduce to you basic concepts of anatomy and things that are still coming in your life. So by mentioning the glomeruli, I've mentioned capillary. Now here's the thing. Capillaries in our bodies, the kidney glomeruli being a great example, are simply blood vessels without the smooth muscle and connective tissue surroundings. So capillaries, where all the fluid exchange takes place in our bodies, are essentially just simple squamous epithelium. Very thin layers, very thin barriers. And so where there are capillary beds, you expect for the body to have the ability to move out of those blood vessels the fluid and nutrient for the cells to live. And in order to do that, those capillaries the simple squamous epithelial cells of those capillaries have to have special ways of doing that in different locations in the body.